Greetings, Brookings AP Bio students. This is Mrs. Rydell, and in this short slideshow, we're going to take a look at the history of some experiments that have to do with our figuring out what the genetic code molecules, that DNA was our genetic code molecule. If you look under your big ideas, essential knowledge 3A, number 1, talks about DNA as the primary source of heritable information. And so we want to look at a couple little videos that are going to help us to see how we came up with that idea that DNA is our genetic code molecule. We're going to start with a little experiment that was done by a medical doctor named Frederick Griffith. And he was looking at strains of bacteria, pneumonia bacteria, that were infecting his patients. And so he was looking at experimenting with those different strains of bacteria to see their effects. And he was using mice to do this experiment. So what he discovered was that when he looked at the cells under the microscope that were the pneumonia bacterial cells, there were basically two kinds of cells. One of them had a very rough looking appearance. He called those the R cells. And the other kinds of cells had a very smooth looking appearance. They had kind of a mucus coating on the outside of the cell that caused them to look very smooth make smooth colonies. And so what he did was to inject those different bacteria into the mice and look to see what happened. What we're going to do is take a look at what happened when he injected these different strains of bacteria into his mice.
Mice injected with R cells remained healthy. When Griffith examined blood samples from these mice, he found no live bacteria.
The S cells, remember, those are the smooth cells.
Mice injected with S cells developed pneumonia and died. When Griffith examined blood samples from these mice, he found live S cells.
So what he did next was to take some of those S cells that killed the mice and heated them up to try and destroy them, and then injected those into the mice. So let's see what happens next.
Mice injected with S cells that were killed by exposure to high temperature remained healthy. When Griffith examined blood samples from these mice, he found no live bacteria.
That doesn't really seem too unusual. You would expect that if you heated and killed the bacteria that they would not be harmful anymore. The interesting part of his experiment comes from this next little experiment that he did. He took some of these heat-killed cells that did not cause the mice to die, and he mixed them with live R cells, which also did not cause the mice to die. And something interesting happened.
Mice injected with a mixture of live R cells and heat-killed S cells developed pneumonia and died. When Griffith examined blood samples from these mice, he found both live R cells and live S cells. Heat killed the S cells, but did not destroy their hereditary material. Somehow, that material was transferred from the dead S cells to living R cells.
There was some sort of material that was passing that told those R cells how to make that smooth coating and become virulent. There was some sort of hereditary material that was passing. That process is called transformation, and we're going to talk a little bit more about that in our slideshow when we talk about bacteria and the genetics of bacteria. But it was the beginning of understanding that there was a molecule that carried the genetic code. Let's take a look at these two experiments now. The Frederick Griffith experiment in 1928 was looking at those two strains of bacteria and seeing ability of the lethal bacteria to pass information to the non-lethal bacteria, even though they were heat killed. There was some kind of a molecule that was passing between those cells that carried the genetic information. Griffith didn't know exactly what that was, but Avery took those experiments one step further in early 1940s by repeating Griffith's experiment, but before he added those heat-killed bacteria to the live strain, he treated them with enzymes to destroy those different molecules. He looked at destroying the RNA, the proteins, the lipids, the carbs, by adding those enzymes and removing those molecules and then looking to see, did the transformation still happen? He was able to tell which of those kinds of molecules was carrying the genetic code. If he destroyed the DNA, the transformation did not happen and the mice lived. If he destroyed these other molecules, as long as the DNA was present, if he destroyed the other molecules, the information was still able to pass to the live bacteria and cause that transformation. It suggested that DNA was our information molecule, was our genetic code molecule. We're going to look one step further, because once we kind of started looking at DNA rather than proteins as our genetic code molecule, scientists actually started looking at DNA. Up until that point, they kind of thought DNA was pretty boring. They really thought that proteins were the genetic code molecule in cells. Proteins are so very different, and lots of different proteins in cells because they do lots of different things. So scientists actually thought that the genetic code molecule must be proteins. DNA seemed pretty boring, not with the repeating A's and T's and G's and C's. It didn't really seem like it could be an information molecule and carry that information. One of the next experiments that helped scientists to start to think about DNA as the code molecule is the Hershey-Chase blender experiment, and we're going to watch a little video about that.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Let's go back to our handout. And remember now, this experiment was looking at viruses that infect bacteria. And by labeling the proteins and the DNA separately with radioactivity, they could tell which molecule was getting into the cell, which one was staying on the outside, and showed that the DNA getting into the cell was the molecule that was carrying the genetic code. One of the first experiments to show that it was DNA, not protein, that was passing on that information. Once we kind of knew that DNA was the genetic code molecule, there was a race on to try and figure out what the structure was, what did it look like, how did this molecule work. And the people who received the Nobel Prize for figuring that out are two gentlemen named James Watson and Francis Crick. And they actually came up with the model that showed the shape, the structure of a DNA molecule. But they did it using photos from another scientist. There was a woman scientist named Rosalind Franklin, and she worked in a lab with another gentleman named Maurice Wilkins, and her lab also was looking at the structure of DNA and trying to figure out what the shape was. What's the shape of the DNA molecule? How did all those A's and T's and G's and C's go together to make a DNA? And she was doing X-ray crystallography, taking X-ray pictures of the DNA molecule and looking at patterns to see if she could figure out what the structure was. Some of her photos, including the famous photo 51, shown in the picture here, were shared by Maurice Wilkins with Watson and Crick, and they used the images from her X-ray crystallography to help them see the pattern and figure out the structure. Later on, they were awarded the Nobel Prize for coming up with the structure along with Maurice Wilkins. By that point, Rosalind Franklin had passed away from cancer, probably from all the exposure to X-rays that she had during the research that she was doing. But she died very young of cancer, and Nobel Prize is not awarded to deceased people, and so she does not share the prize for figuring out the structure, even though we know her work, her research was instrumental in allowing those other gentlemen to figure that out. We also mentioned Erwin Chargaff in the 1940s. He analyzed DNA from different organisms and helped to provide some beginning information about how the DNA molecule went together by looking at those relationships between the nitrogen bases. And remember Chargaff's rule, A's always equal T's, the concentration, the amount of A's in a DNA molecule always equals T's, and G's always equals C's. That made a lot more sense once we figured out the structure of DNA and how it goes together. We're going to talk a little bit more about some of the other experiments that helped us understand DNA a little better. And the next experiment we're going to look at was one done by Messelson and Stahl in the late 1950s. After we figured out the structure of DNA, understanding how replication worked was kind of the next step. And so we're going to watch a little video about how that worked.
The Messelson and Stahl experiment provides evidence for semi-conservative replication of the DNA molecule, where the two parent strands serve as the template for synthesis of new strands. In this experiment, bacterial cells were grown for several generations on a medium containing a heavy isotope of nitrogen, N15. The DNA in these cells therefore contained heavy N15 nitrogen. The cells were then transferred to a new medium containing the normal lighter isotope, N14. At various times after the transfer, samples of the bacteria were collected. The DNA was then extracted and dissolved in a solution of cesium chloride. The samples were then spun rapidly in a centrifuge. When the cesium chloride is centrifuged at high speed, a concentration gradient is established in the tube. DNA molecules move in the gradient until they reach a place where their density equals that of the cesium. DNA containing N14 moved to a position in the gradient determined by its density. DNA containing N15 is denser than that containing N14, so it sank to a lower position in the cesium gradient. After one generation in N14 medium, the bacteria yielded a single band of DNA with a density between that of N14 DNA and N15 DNA, indicating that only one strand of each duplex contained N15. After two generations in N14 medium, two bands were obtained, one of intermediate density, in which one of the strands contained N15, and one of low density, in which neither strand contained N15. Messelson and Stahl concluded that replication of the DNA duplex involves building new molecules by separating parent strands and then adding new nucleotides to form the complementary strand on each of these templates.
Remember, we talked about the three possible patterns for replication when we did our replication with DNA and looked at the very first chapter. And we looked at some possible models. The semi-conservative is the one that actually is the way DNA copies itself, splitting the strand, using the two separated strands to act as templates to fill in with a new matching complementary strand. And so each of the final DNA copies ends up with one old strand and one new strand. That's the pattern, and it was determined by those experiments with Mendelssohn and Stahl. The other possibility was to keep the strand together and use that as a template or a model to make a second strand that is an exact copy. In this case, both of the strands in this molecule are going to be the old strand, and both of the strands would be new strands in the copy. That's called the conservative method, and that is not the way DNA copies itself. The other possibility was to copy in sections and have combinations of old and new together, not individual strands, old and new, but sections of DNA, old and new, interspersed. And the experiments that Mendelssohn and Stahl came up with, looking at how DNA copies itself, the patterns that we saw allowed them to figure out that the semi-conservative method was the way of that those DNA molecules copied themselves. We're going to do a little activity looking at this a little bit closer in later on. The other experiment, way earlier than that, early 1900s, we talked a little bit about this when we talked about our genetic disorders, is Garrett's idea of inborn errors of metabolism, that genetic diseases produce changes in enzymes that cause genetic problems. And he was looking at patients, he was a medical doctor, and looking at patients that had that black urine disease. We talked about that in our other slideshow. If you are missing an enzyme that does a chemical reaction, those molecules are not going to be broken down. And he was suggesting the idea that those metabolic disorders, if you have a missing enzyme, not being able to do some kind of a chemical reaction is going to lead to disease. And it was kind of the first idea that showed that genes suggest that genes, the genetic code molecule, at that time they didn't really know what it was causing a change in phenotype, and that enzymes were involved in catalyzing specific chemical reactions, and changes in a code can result in genetic disease. We also talked a little bit about the Beadle-Tatum experiment in our last slideshow, and the idea that creating mutants, different enzyme mutants, and working out the pathways of how these enzymes work together to make a pathway, and a chemical pathway in the cell, and that blocking enzymes along the way cause changes in the pathway that are going to produce phenotype problems, and could be the cause of a genetic disease. And they worked out the pathway of a lot of the amino acids that are important for growth by examining, creating mutants, and then seeing what enzymes they were missing. And that kind of led to that idea of one gene producing one enzyme. The one gene, one enzyme hypothesis has since changed a little bit, and we talked about that in our last slide as well. One gene, one polypeptide, we're making a protein chain. Now we know that not all genes code for proteins, some code for RNAs. There are other kinds of DNA code in our DNA sequence that's not making enzymes, and so we've kind of come up with a modified version of that one gene, one enzyme. Maybe one gene makes one polypeptide or one RNA molecule. And we talked also a little bit about Nuremberg's idea of figuring out what the genetic code was by creating artificial mRNAs, and then looking to see what kind of amino acid sequences they produce by combining ribosomes and nucleotides in test tubes outside of the body, and making artificial code, and then seeing what proteins get made from that. Thanks for watching, and stay tuned for more activities with DNA.